Okay, so this is going to be chapter 18, which is infections of the skin and eye. So your skin is the integument, which is actually one of the largest, it is the largest organ in the body. It's also one of your primary surface barriers, protecting you from the outside world. Here you can see some basic background on the skin as far as the size. It also does include your hair, nails, sweat, and oil glands as well. So the stratum corneum, which is the outermost layer of the skin, is packed with keratin. And this actually gives the cells the ability to withstand damage and abrasion. It's actually a very tough protective barrier. And the keratin actually is very tough for certain bacteria or fungal species to be able to break through. And as you'll see, most infections of the skin that originate as either bacterial or fungal usually have to be introduced due to damage to the skin, either a cut, a surgical site, something like that. Lipids are packed between the cells to give the stratum corneum a water repellent property. Now, the entire epidermis is replaced about every 25 to 45 days. And this is because you have in the stratum basale, which is the base layer of the skin, stem cells that are just constantly undergoing mitosis. Now, the dermis, this is composed of connective tissue. It's a rich matrix of fibroblast cells and fibers such as collagen. You will have some macrophages and mast cells, so you will have the presence of some immune cells. The dermis will also harbor a dense network of nerves, blood vessels, and lymphatic vessels. And when you actually do cut the skin or break the skin and you actually have bleeding, that is actually, you have passed the epidermis and have entered into the dermis. The epidermis is avascular, so there's no blood vessels in the epidermis, but there's definitely blood vessels in the dermis. Blister formation, this results of friction trauma or burns to the dermis, and it causes a separation between the dermis and the epidermis. And here we can see the two different layers of the skin. Here you have multiple layers of flat cells that are keratinized. This is your epidermis right here. And then this whole area here would be your dermis. And then down here in the yellow, you have what's called the subcutaneous layer, which is actually not part of the dermis, or not part of the skin, but it's actually just underneath the skin. As far as microbial growth on the skin, we'll talk about this in a few minutes, but as you can see, there's not an abundance of organisms in this diagram on the skin, and that's mainly because it's not a very attractive place to live. We'll talk about why in a few minutes. As far as your hair follicles, the roots of hairs are located in the dermis. You also will have sebaceous or oil glands that will are associated with the hair follicles. You have sweat glands, which are separate from the follicle, and they're located in the dermis as well. And all glands will have openings onto their surfaces, which pass through the epidermis. So some of your defense mechanisms that can play a role here are what we call antimicrobial proteins or antimicrobial peptides. They've been identified over the past 2 decades, and they are positively charged chemicals that act by disrupting negatively charged membranes of bacteria. There are many, many different families of antimicrobial peptides. For example, you have what are called histatins or defensins, to name a few. These are actual proteins that are synthesized and secreted by your cells, and they have antimicrobial properties. These antimicrobial peptides are responsible for keeping the microbial count on the skin relatively low. Now, sebum, which is secreted by the sebaceous glands. This actually creates a low pH, and this low pH, what we call the acid mantle, makes the skin inhospitable to most microorganisms. They don't like the low pH. The high concentration of lipids serves as nutrients for normal microbiota. And the breakdown of fatty acids leads to toxic byproducts that inhibit the growth of other microbes that are not adapted for living on the skin. Sweat, this also contributes to the acid mantle or the low pH. But more importantly, sweat's also going to bring a very high concentration of salt. And many, many bacteria actually are not able to tolerate any moderate to high concentrations of salt. Now, you'll see in a few minutes that there is a species that kind of loves salt, and that would be your Staphylococci species. And Staphylococci species are the most common bacteria that we find on the surface of the skin. You can also have micrococci species, such as M. luteus, which is one of the strains that you guys are using in the lab. But like I said, staph is really the primary player here. And we'll look at that in a few minutes as well. Lysozyme. This is an enzyme that's found in sweat, tears, and saliva. And it actually, lysozyme will break down peptidoglycan, essentially destabilizing the cell wall, causing the bacteria to die. So this is actually, it's a protein that's produced by the bacterial cell. Again, that also plays another role in having antimicrobial properties. All right, so normal biota of the skin. Overall, the skin is not a very attractive place to live. It is vast, it is dry, it's a very salty surface, and it's coated with a lot of antimicrobial proteins as well as antimicrobial lipids. More dense microbial populations in moist areas and skin folds, and in protected environments of the hair follicles and glandular ducts, because these are much more attractive areas to live. Your mucosal surfaces are an ideal place for most microorganisms. The skin, not so much. Now, hundreds of species of microbes are found distributed over many different areas of the body. And there are 5 major taxa represented in the microbiota. This is what the Human Microbiome Project has sort of given us so far. Predominance of these groups is varied in the different regions sampled. Large differences among people with respect to the types of microbes found in various skin sites. An individual's own skin microbiota seems to be relatively stable over time, though. Staph epidermidis and Propionibacterium acne are found in large numbers on the skin, again, primarily due to their ability to tolerate that high salt. About 4% of the population will carry Staph aureus, as well as Staph epidermidis. So if we look at the skin, our defense mechanisms for the skin are keratinized surfaces, constantly sloughing off the layers of the skin, low pH, high salt, and these antimicrobial proteins. Normal microbiota, staph species, Propionibacterium species, which causes acne. Then you can have specific species like yeast, like Candida species, that can be found on the surface as well. Okay, so let's talk about one of infections, and this is something that probably everybody has heard of. It's called MRSA. MRSA stands for methicillin-resistant Staph aureus. It's a common cause of skin lesions in non-hospitalized people. It's resistant to multiple antibiotics. As you can see here, this is actually an antibiotic test. Essentially what we did here was we took a strain of staph and we just took a swab of it and just kind of plated it all over the plate. We then applied these little drugs, disc, and these little white discs they're saturated with drug. And once the drug actually hits the agar, what will happen is the drug will diffuse into the agar. And the further away from the disc, the less concentration of drug. So if the drug actually has an effect on the bacteria, you'll see what we call a zone of inhibition. What this is telling you is that the drug is actually inhibiting the growth of the bacteria. Okay. This is vancomycin. It's a very powerful drug. It's usually the antibiotic of choice in the emergency rooms, especially if they don't know what's causing any type of particular infection. Then we have methicillin, penicillin, and erythromycin. And what you notice here is the bacteria grew all the way up to the disc. That tells me that this drug has absolutely no effect on the bacteria. And the reason why it has no effect on the bacteria is that it is a MRSA strain. And just because you're resistant to methicillin doesn't mean that's the only drug you're resistant to. Chances are you're resistant to multiple antibiotics, as shown in this example here. Infections of MRSA tend to be raised, red, tender, localized lesions, often feature pus and feel hot to the touch. And they occur in breaks of the skin from injury, shaving, or abrading. Now, most MRSA infections actually are acquired in the hospital. And these are a little more serious, and we'll talk about these when we get to infections of the cardiovascular system and blood, because if you pick up a MRSA infection in the hospital and it becomes a bloodstream infection, that can be quite, you can have some complications there. So we will see that again. But as far as the soft tissue infections on the skin, like I said, this is kind of like what you're dealing with here. So what is Staph aureus? Well, Staph aureus is a gram-positive cocci that forms grape-like clusters. Remember, staph means clusters and cocci means circles from chapter 4. They are non-motile. They are the sturdiest of all non-endospore-forming pathogens. And one of the interesting features is that they can actually withstand high salt extremes in pH and high temperature. Okay. And what you're looking at here is in what looks like a bundle of grapes there. That's actually a scanning electron micrograph of the Staph aureus cocci. And the plate that you're looking at is mannitol salt agar. And this particular plate is both a differential and selective media for the growth of staph species. Unlike many organisms, Staph aureus can withstand relatively high concentrations of salt. And it's able to grow on a mannitol salt agar plate, which has very high concentrations of salt. In addition, it can ferment mannitol, which is a sugar found in the medium, leading to a yellow color change over time. So what you're looking at here is, the initial streak out right here, grows up for a little more time, and then fully grows up. But what you're also seeing is a change in the color of the medium, telling you that not only can this grow on a mannitol salt agar plate, but it's also able to ferment the sugar mannitol, which is a defining feature of Staph aureus. Another very common staph species is Staph epidermis. It will also grow on an MSA plate, but it will not ferment mannitol. Okay, so that would remain pink or reddish color. And with the mannitol salt agar plate, this is one of the plates that you will actually be using as one of your differential and selective medias. You also have, if we look at a blood agar plate, now typically blood agar plates are made with sheep's blood agar. And the blood agar plate is growing Staph aureus here. Some strains show 2 zones of hemolysis caused by 2 different hemolysins. The inner zone is clear, where the outer zone is fuzzy, and it appears as if only the plate has sort of been refrigerated after growth here. So you have this inner zone of hemolysis, and then you sort of have this outer zone, and then you sort of have, the regular plate here. This is a very common, phenotype of Staph aureus on a blood agar plate. Another test that can be done is what we call a coagulase test. Coagulase is an enzyme that will cause clumping of the bacteria. And what you can do is take plasma that is inoculated with staph bacteria. So for example, like a bloodstream infection, or you can even take some scrapings or pus off the surface and mix it with coagulase. If the reaction does not clump up and it's a negative reaction and therefore it's coagulase negative, it's staph epidermidis. If it does clump up and it's a positive coagulase test, then it is Staph aureus. And this is important because, physicians kind of want to know, are they dealing with Staph epidermidis, which is very easily treated, or are they dealing with Staph aureus, which could potentially have, the opportunity for lots of drug resistance to sort of play a role there. Okay, so here's just a little summary table. We're looking at methicillin-resistant Staph aureus. Common modes of transmission are direct or indirect contacts. Prevention is just really washing your hands. Treatment would be vancomycin. Remember, in this particular drug resistance slide here, we did see that vancomycin is usually quite effective at treating this. It is considered a serious threat in the CDC's antibiotic resistance report, which came out in 2020. This is, an organism, especially the drug-resistant strains, that we are concerned about. Community-associated MRSA infections are most common in children and young to middle-aged adults. It's usually a result of contact sports. Most infections in the US are due to hospitals, but we are seeing that come down with, better practices. Okay, Impetigo, this is a superficial bacterial infection that causes the skin to flake or peel off. It's caused by either Staph aureus or Strep pyogenes, or a mixture of the two. Children are the primary victims. Symptoms are the same regardless of the organism that's causing the infection. Now, kind of heading back here, Staph aureus does have some virulence factors. Virulence factors are some type of protein product that's produced by the bacteria that can cause infection, that can cause what we call disease or pathogenesis. Virulence factors, like I said, they're oftentimes some type of protein factor like an enzyme or a toxin that gets secreted from the cells. And oftentimes those enzymes or toxins will do tissue damage to the host. And Staph aureus will release exotoxins called exfoliative toxins A and B. And what they do is they attack a protein that is important for epithelial cell-to-cell binding. Breaking this protein leads to the characteristics of blistering. They also have coagulase. Coagulase will coagulate plasma and blood, causing fibrin to be deposited around the bacteria. And what this will do is it will concentrate the exotoxin in the area of local damage. Now, Strep pyogenes, this is an organism that also causes strep throat. So we will see this guy again when we get to the respiratory system. It can cause scarlet fever, pneumonia, necrotizing fasciitis, and various types of bloodstream infections and rheumatic fever. Strep pyogenes is a gram-positive cocci, much like Staph aureus is. It is beta hemolytic on a blood agar plate. So beta hemolysis means that it will undergo complete hemolysis of the blood or breakdown of the blood. And like Staph aureus, it does possess some of these exotoxins here. And here we have an identification test for Strep pyogenes. And what you're looking at here in A, this is a rapid direct test kit for the diagnosis of group A infections. With this method, what you do is you'll take this, say, a swab of the patient's throat, and then you'll mix it with some latex beads that have monoclonal antibodies, and a positive reaction, if it occurs, you'll see visible clumps. And that's an indication because the monoclonal antibodies are specific for strep pyogenes. If you see that coagulation is clumping, as you see right here, that's a positive test. If there's no clumping, then it's a negative test. Okay. On the blood agar plate here, we have a bacitracin disc, which is a drug disc here. Okay. With very few exceptions, only strep pyogenes is sensitive to a minute concentration of bacitracin. Any zone of inhibition around the drug disc here is interpretive as a presumptive indication for a positive for strep pyogenes. And if you look here, you can see you have this little zone of inhibition around the bacteria right here. Okay, so looking at impetigo diseases, again, it can be caused by Staph aureus or strep pyogenes. And here you can see the mode of transmission. Prevention for both of these is good hygiene practices. And you can see the treatment mechanisms as well. Okay. Cellulitis, this is caused by a fast-spreading infection in the dermis and subcutaneous tissue. It causes pain, tenderness, swelling, and warmth. It can lead to a fever and swelling of the lymph nodes. It's frequently displayed as red lines leading away from the affected area. It's very common in the lower extremities, and people with immune-compromised systems or have cardiac insufficiency are a little more at risk for cellulitis. Now, what can cause cellulitis? MRSA, staph strain, Staph aureus, and then also strep pyogenes. Okay, and you can sort of see the breakdown here. Okay, I am actually going to stop here because I have to run to go give a live lecture, and I will pick up with recording too.